Anergy is a term in immunobiology that describes a lack of reaction by the body's defense mechanisms to foreign substances, and consists of a direct induction of peripheral lymphocyte tolerance. An individual in a state of anergy often indicates that the immune system is unable to mount a normal immune response against a specific antigen, usually a self-antigen. Lymphocytes are said to be anergic when they fail to respond to their specific antigen. Anergy is one of three processes that induce tolerance, modifying the immune system to prevent self-destruction, the others being clonal deletion and immunoregulation. Mechanism. This phenomenon was first described in B lymphocytes by Gustav Nossel and termed clonal anergy. The clones of B lymphocytes in this case can still be found alive in the circulation, but are ineffective at mounting immune responses. Later, Ronald Schwartz and Mark Jenkins described a similar process operating in the T lymphocyte. Many viruses, HIV being the most extreme example, seem to exploit the immune system's use of tolerance induction to evade the immune system, though the suppression of specific antigens is done by fewer pathogens, notably Mycobacterium leprae. At the cellular level, anergy is the inability of an immune cell to mount a complete response against its target. In the immune system, circulating cells called lymphocytes form a primary army that defends the body against pathogenic viruses, bacteria and parasites. There are two major kinds of lymphocytes, the T lymphocyte and the B lymphocyte. Among the millions of lymphocytes in the human body, only a few actually are specific for any particular infectious agent. At the time of infection, these few cells must be recruited and allowed to multiply rapidly. This process, called clonal expansion, allows the body to quickly mobilize an army of clones as and when required. Such immune response is anticipatory and its specificity is assured by pre-existing clones of lymphocytes, which expand in response to specific antigen. Process called clonal selection. This specific clonal army then combats the pathogen until the body is free of the infection. Following clearance of the infection, the clones that are no longer needed die away naturally. However, a small number of the body's army of lymphocytes are able to react with proteins that are normally present in a healthy body. The clonal expansion of those cells can lead to autoimmune diseases, wherein the body attacks itself. In order to prevent this process, lymphocytes possess an intrinsic quality control mechanism. This machinery shuts down the lymphocyte's ability to expand if the trigger for the expansion turns out to be the body's own protein. T-cell anergy can arise when the T-cell does not receive appropriate co-stimulation in the presence of specific antigen recognition. B-cell anergy can be induced by exposure to soluble circulating antigen, and is often marked by a downregulation of surface IgM expression and partial blockade of intracellular signaling pathways. Molecular mechanism of anergy induction in T lymphocytes. Stimulation of the T-cell receptor, TCR, along with co-stimulatory receptors of a T lymphocyte triggers balanced activation of all the T-cell signaling pathways. Full T-cell stimulation. In this case, beside other pathways, calcium-dependent arm of a lymphocyte signaling is activated by TCR. This leads to an elevation of intracellular Ca+E concentration. Under this condition, calcium-dependent phosphatase calcineurin removes phosphates from a transcriptional factor NFAT, which in turn translocates to the nucleus. Additionally, during full T-cell stimulation, a co-stimulatory receptor CD28 activates PI3K or other pathways that eventually lead to increased nuclear levels of REL, NF-kappa B and AP1 transcription factors. much more than just by the TCR activation alone. AP1. Phosgene heterodimer further heterodimerizes with NFAT, forming a transcriptional complex which promotes transcription of T-cell productive response-associated genes. Those are, for example, IL-2 and its receptor. On the contrary, TCR signaling without costimulatory receptors sufficiently activates only the calcium arm of the signaling, leading only to the activation of NFAT. However, without the necessary induction of AP1 by other pathways, activated NFAT is unable to form the transcriptional complex with AP1, as it does during complete T-cell activation. Productive response. In this case, NFAT homodimerizes complexes with itself, working as a transcriptional factor that induces anergy in the lymphocyte instead. NFAT homodimers are directly responsible for the expression of anergy-associated genes such as ubiquitin ligase GRAIL or a protease caspases 3. Moreover, the expression levels of IL-2, but also, for example, TNF-alpha and IFN-gamma, typical for productive response, are actively decreased in the anergized cell. Anergized cells tend to produce anti-inflammatory IL-10 instead. There are 3 NFAT proteins in the T-cell: NFAT1, NFAT2 and NFAT4, and apparently are redundant to some extent. Thus, when an antigen is properly presented to the T lymphocytes by an antigen-presenting cell, APC, which displays the antigen on its MHC2 complex and which activates T-cell's costimulatory receptors, T lymphocytes undergo productive response. However, when T-cells interact with an antigen not presented by the APCs, that is very probably not the antigen that an immune response should be held against. The T-cell undergoes anergy. It has also been shown that certain antigens properly presented by the APCs induce the T-cell activation only weakly. This weak stimuli still activates NFAT sufficiently. However, AP1 is not. Thereby the anergistic response takes place even with the costimulation. Strong stimulation of T-cells either by IL-2 or by TCR costimulatory receptors can break the anergy. Clinical significance. Anergy may be taken advantage of for therapeutic uses. The immune response to grafting of transplanted organs and tissues could be minimized without weakening the entire immune system. A side effect of immunosuppressive drugs like cyclosporine. Anergy may also be used to induce activated lymphocytes to become unresponsive with autoimmune diseases like diabetes mellitus, multiple sclerosis and rheumatoid arthritis. Likewise, preventing anergy in response to a tumoral growth may help in anti-tumor responses. It might also be used for immunotherapeutic treatment of allergies. Dominant tolerance. Dominant and recessive tolerance are forms of a peripheral tolerance. The other tolerance beside peripheral is a central tolerance, where so-called recessive tolerance is associated with anergized lymphocytes as described above. In the dominant form of tolerance, specialized Treg cells which actively ablate the immune response are developed from the naive T lymphocyte. Similarly to recessive tolerance, unopposed NFAT signaling is also important for Treg induction. In this case, the NFAT pathway activates another transcription factor, FOXP3, that is a marker of Tregs and participates in the genetic program. Testing. The multitest Meriu, or CMI multitest system. Multitest IMC. Istituto Maria Italia, Rome, Italy, has been used as a general test of the level of cellular immunity. It is an intradermal test of skin reactivity, similar to tuberculin tests, in which a control, glycerol, is used with 7 antigens of bacterial or fungal origin: tetanus toxoid, tuberculin, diphtheria, streptococcus, candida, trichophytin, and proteus. In this test, reactions are categorized according to the number of antigens provoking a response, and the summed extent of the skin response to all 7 antigens. Here anergy is defined as a region of skin reactivity of 0 to 1 mm. Hypergy is a reaction of 2 to 9 mm in response to fewer than 3 antigens. Normergic is a reaction of 10 to 39 mm to 3 or more antigens. And hyperergy for a reaction of 40 mm or more. Experimental approaches to study anergy. Various chemicals inducing, inhibiting described T-cell signaling pathways can be used to study the anergy. The anergy in T-cells can be induced by ionomycin, the ionophore capable of raising intracellular concentration of calcium ions artificially. Conversely, Ca plus E chelators such as EGTA can sequester calcium ions, making them unable to cause the anergy. Blocking of the pathway leading to the anergy can be also done by cyclosporin A, which is capable of inhibiting calcineurin, the phosphatase responsible for dephosphorylating of NF-AT, priming its activation. PMA. 4-bul-12-myristate 13 acetate, along with ionomycin, is used to induce full T cells activation by mimicking signals provided naturally by TCR, co-stimulatory receptors activation. Further reading. Jenkins, Mark A. February 1992. The Role of Cell Division in the Induction of Clonal Anergy. Immunology Today. 13. 2. 69-73. PMID 1349483. DOI 10.1016/0167-5699. 92. 90137V. Clonal Anergy at the U.S. National Library of Medicine. Medical subject headings. MESH.